Welcome everyone to today's session of the Wednesday afternoon lecture series here at the NIH. My name is Anirban Banerjee. I'm a senior investigator in the intramural program of NICHD, and today's lecture is hosted by the Metals in Biology Scientific Interest Group. It's a real honor and a great pleasure to introduce today's speaker, who's really our next-door neighbor, Professor Iqbal Hamza, a professor of pediatrics at the Center for Blood Oxygen Transport and Homeostasis at the University of Maryland School of Medicine. Iqbal grew up in India, in Bombay, to be more specific, for most of his formative years, and received his BSc and MSc degrees in biochemistry from the University of Bombay. He then came over to the US, to the State University of New York at Buffalo School of Medicine campus, in the lab of Mark Bryan, where his work elucidated the role of bacterial iron response regulator in coordinating iron homeostasis and heme synthesis. And I suppose this is when sort of the bug of metal biology bit Iqbal, because he then went on to do a postdoctoral research in the labs of Jonathan Gitlin, where he expanded his tool chest to transgenic mice and exploited mammalian cells to model human genetic disease of copper metabolism, specifically Menkes and Wilson's disease, and established the molecular and genetic basis for copper trafficking by metal chaperones. And in Iqbal's own words, empowered with a 3-week crash course in C. elegans genetics at the Cold Spring Harbor laboratory, he started at the University of Maryland, where he embarked on a completely new direction, that of heme trafficking pathway in eukaryotes. So there are probably a lot of useful nuggets of wisdom for those of you postdocs who are looking to start your lab. His choice of system was exceptionally precise, because it turns out that C. elegans does not utilize heme, but actually utilizes does not make its own heme, but utilizes environmental heme. And with this system, Iqbal's lab, they have gone on to make pioneering discoveries in heme transport and trafficking, and in turn have established connections to a number of human diseases, some of which I hope he's going to touch upon today. Iqbal has received numerous awards for his work, including the NIH Research Career Development Award, the Junior Faculty Excellence Award, and the Kirwan Faculty Research and Scholarship Prize at the University of Maryland. He's a fellow of the Japan Society for the Promotion of Science, and a fellow of the American Association for the Advancement of Science. But more importantly, Iqbal actually has been a particularly strong mentor of young scientists in the area of metal biology, not only in his own department, but outside in the entire community. And it's really very highly appreciated because of that. He's had a long interaction with the NIH intramural community, and we look forward to these continuing interactions. Thanks very much for accepting our invitation, Iqbal.
It's really an honor and a pleasure to be here. It's not the fact that I'm at the NIH WALS, and that truly is an honor, but the fact that I know half of this audience, right? The joke aside, one of the best things of being at the University of Maryland is the fact that it's so close to the NIH, where I have some amazing people that I can collaborate with or just bounce ideas with. And few of them are sitting right here in the audience. What was not clear from Anirban's introduction is that 2 of my sabbaticals, 2010 and 2019, were done at the NIH. The first one with Paul Liu at NHGRI, and the second one with David Bodine, also at NHGRI. You'll also see that there is a theme in the talk today, and that is the power of using model systems to uncover unanswered questions. And you'll see that. And the use of those organisms shown here is C. elegans, along with a whole series of different model systems. So our fundamental questions are to understand the biology of heme. I don't fit in as a microbiologist, a hematologist, or a parasitologist. Our fundamental questions is simple. How does heme, as you'll see in the next slide, a hydrophobic, cytotoxic, cyclic organic ring, moves around between cells and within cells? To be able to answer that, we use C. elegans, and the ultimate goal is, of course, to be able to uncover these pathways and the implications of those pathways in human health and disease. To go from C. elegans to humans, we have to cross many different barriers, and we overcome those barriers using many different model systems. As Anirban said, I did my PhD doing bacterial genetics, and then I moved to mouse genetics. So anywhere from bacteria to man or to mouse, there are various other organisms that you can transcend. And each of those organisms that you use can be exploited for its specific purpose that it brings to the genetic toolbox. Okay. We use yeast, we use zebrafish. After my first sabbatical, I set up a fish facility. Mouse is where I had gotten my postdoctoral training. So we have a large mouse colony, and something that I did not anticipate 20 years ago when I studied at the University of Maryland is our interest in parasitology. So now a third of my lab works on interesting parasites, including leishmania. So at the end, if you look at the scale bar, you can see that the scale bar between mice and worms are almost the same, because for us, the worm is as large as a mouse in terms of its genetic power and its implications in human health and disease. So the reason why I put this slide up is because people often confuse heme with globins. Hemes are not just for globins. As somebody else told me, if you're studying heme trafficking, why don't you just put a GFP tag on the heme, right? So this is a misnomer because people often confuse heme with globins. Here is the structure of heme. It's typically a planar ring. It's got hydrophobic side chains, which makes this molecule hydrophobic, which can intercalate into biological membranes and causes cytotoxicity because of the ion in the middle, which generates hydroxyl radicals, and the porphyrin ring itself catalyzes reactive oxygen species generation. In here is the first molecule that has been fully characterized, and over 400 of these class of molecules called globins have been crystallized. Their biophysical and biochemical properties have been elucidated. But here's the interesting question. We don't know how heme gets into globin after 70 years. And this is really a phenomenally open question. These are enormous gaps in our knowledge. So hemes are not simply for globins, but they're also involved in other kind of interesting biological functions, such as synthesis of nitric oxide, so NOSes, heme-containing enzymes, heme sensing proteins such as soluble guanylyl cyclases are heme enzymes, catalases, which are involved in detoxifications, and P450s, prostaglandin synthesis require heme. P450 side chain cleavage enzymes are involved in sterol metabolism. Cytochrome Cs and cytochrome C oxidases in ATP synthesis and OXPHOX reactions. Hemes are involved in producing pus in neutrophils, myeloperoxidase. And hemes are involved in the conversion of T3 to T4. So hemes perform really diverse biological functions. And in all of these cases, hemes are prosthetic groups, which means that they sit at the catalytic site and do interesting chemical reactions. But a new class of proteins have been recently discovered in the past decade, where hemes are not simply static prosthetic groups doing catalytic functions, but they're really cofactors which are involved in controlling biological functions as signaling molecules. So heme binding to transcription factor mPER or Weber alpha controls circadian clock and couples it with metabolism. Heme binding to DGCR8 allows how much pre-pri mRNA or microRNA is produced. Heme binding to the slow BK potassium channel or the epithelial sodium channel allows it to open and close quicker or slower. And heme binding to cystathionine beta synthesis allows how much hydrogen sulfide to be produced. So one thing is clear from this slide, that heme is at the intersection of four gases that has physiological roles: oxygen binding, nitric oxide sensing and synthesis, hydrogen sulfide. And if you degrade this heme molecule by breaking one of the methane bridges, you open up the ring, you release iron, but you also produce carbon monoxide. And I'll come back to that later. Okay. So this is the question that I got interested 21 years ago when I was still a postdoc at Washington University. Heme is made in the mitochondrial matrix of eukaryotic cells. It's made through a highly defined biochemical pathway shown here that is spatially separated between the mitochondria and the cytosol. These are 8 invariable enzymes with 8 genes that are highly conserved across organisms. In plants, the first step is through a tRNA intermediate. So the iron enters in the last step of heme synthesis, where the ring that you saw in the preceding slide gets iron inserted through a catalytic reaction by an enzyme called ferrochelatase, and then you make that heme that you saw in the previous slide. It's made in the mitochondrial matrix. And the question that I got really interested in is, how does a hydrophobic molecule that is cytotoxic get out of the mitochondrial inner membrane to be put into cytochrome Cs and all the heme-containing proteins that I gave you examples of? How does heme get into globins, guanylyl cyclases? How does it get into the nucleus to be able to de-repress genes by binding to transcriptional repressors? How does heme get into prostaglandin synthases that are inside the ER lumen? How does heme get into peroxidases in the lysosomes or catalases in the peroxisomes? The reason why this question has been so difficult to answer is because of heme synthesis itself, which is a highly complicated and regulated biosynthetic pathway. It is regulated by the amount of iron that's available. It's regulated by the flux of these intermediate steps, because these intermediate products or substrates are highly toxic because they can generate reactive oxygen species. And hemes are itself toxic. So you have a toxic substrate, you have a toxic intermediate, and you have a toxic product that is made, but it's absolutely crucial for life. Okay. So therefore, heme synthesis is highly coupled with apoprotein synthesis and regulated by factors such as iron, heme itself, and gases. So how does one spatially separate synthesis from trafficking? That was the problem. Okay. So as I got interested in the question, my colleague Jan Abkovitz uncovered a protein which she named feline leukemia virus cell receptor. This protein, which is actually a 12 transmembrane domain protein, binds heme and is able to export heme. And the way she uncovered it was showing that cats that are infected with feline leukemia virus get something called pure red cell aplasia. The reason for this is because when the virus infects, it binds to this protein, internalizes it and degrades it, and causes apoptosis of red cells because they're unable to export heme out. While this was a decades-long inquiry into how feline leukemia virus causes red cell aplasia, I was more interested in a genetic approach to separate synthesis from trafficking. So when I was still a postdoc at WashU, I was really considering, how do I go about answering this question? So here are the 7 steps of heme synthesis. I have not put the first enzyme because they are different between plants and non-plant organisms. So as you can see from the blast value mice, flies, yeast, I could put E. coli there. They're the same heme biosynthetic pathway enzymes. Okay. If you look at the C. elegans genome, we could not find any homologs of the heme synthesis enzyme. Now this was really interesting, because when Bob Waterston published the C. elegans genome in 1997, right, none of these genes were identified. It's possible that they missed a part of the chromosome. It is also possible that C. elegans doesn't make heme but doesn't care for it, such as some organisms, microorganisms such as Haemophilus influenza. The other possibility is that C. elegans makes heme through a completely novel pathway, which in of itself would be very interesting. Or the fourth possibility was that C. elegans does not make heme, but acquires it exogenously. So this is where I took a 3-week course at Cold Spring Harbor, because I said maybe C. elegans is the organism which we can spatially separate our synthesis from trafficking. So after taking the course, I realized that C. elegans eats bacteria, E. coli in the lab, and E. coli has the heme biosynthetic pathway the same way that you and I do. So how can one grow worms by still controlling the levels of heme? So I was able to send my American postdoc to the US military base at Fort Detrick because the US Army was interested in using C. elegans, and they had developed an azenic liquid culture, which they named CHR. So when she came back to the lab, I couldn't get onto the military base at that time because I was still an Indian citizen. And I said, what is in the liquid synthetic medium? And the liquid synthetic medium had cytochrome C. If you take out cytochrome C from this liquid culture, the worms don't grow. And if you start adding pure hemin, they grow really well. They have an optimal heme requirement of 20 micromolar. And as you increase the concentration of the heme, worm stops growing, and eventually it becomes really cytotoxic. So this was a year after I started my lab at Maryland, still trying to uncover whether C. elegans makes heme or acquires heme exogenously. Once we did this, now we could do some really interesting biochemical experiments, including with radiolabeled heme, to show that indeed C. elegans does not make heme, but get it exogenously. So here is heme biosynthetic enzyme activities, the second, third, and the last enzyme. None detected for Cinorhabditis elegans. And then, since we are in close proximity to the USDA, I was able to get to the hematology lab and looked at other free-living nematodes. None of them we could detect enzyme activities. And then we went to the animal hematology laboratory. And this is a huge industry. Parasitic nematodes, also called helminths, right? So we have your threadworm, hookworm, barber poorworm, whipworm, and roundworm, which infects humans, livestock, and plants. None of them we could detect heme enzyme activities. This is pre-genome era. So now we do know that C. elegans and related helminths are all heme auxotrophs. They have lost all 8 genes for heme synthesis. So if you have not been to a worm lab, I'm telling you that it is an extremely powerful model system for us specifically. Small size, a lot smaller than mice. You can do some powerful genetics, optically translucent from an egg to an egg-laying adult. Short life cycle, 3.5 days. Each hermaphrodite shown here lays anywhere between 200 to 500 progeny, and males occur due to chromosomal non-disjunction, one in every 500, and they're rare but useful for genetic crosses. Superb forward and reverse genetics, which we're going to discuss today. The first animal genome to be fully sequenced in 1997. Every somatic cell lineage has been mapped by Sulston et al. Okay. The entire animal has been sectioned from the mouth to the tail at the EM level, ultrastructural level, right? And for us, the most powerful thing for us, that we can grow them azenically on a large scale to do interesting metabolism and biochemical studies, because it's a natural mutant for heme synthesis. So it's phenotypically and genotypically normal for it to not make heme, but acquire heme from the environment. So since C. elegans is an animal, a hermaphrodite has 959 cells. It has 20 beautiful polarized intestinal cells through which it eats, and it has absorptive and storage functions. So here is a single cartoon of a polarized intestinal cell. This is the lumen of the intestine. Okay. Since it lacks all 8 genes for heme synthesis, it takes up heme from its food and directly incorporates it, as we have shown using radiolabeled tracer experiments, into heme proteins, into cytochrome oxidases. It has 5 guanyl cyclases. we have only 4 globins. They have 33 different globins for which we don't really understand their function. They have all the transcription factors which binds heme. They have over 80 P450 enzymes. They're peroxidases and catalases. So we have complete control over the flux of heme, all within the context of a transparent animal. And more importantly, not only do they have to acquire heme and incorporate in toto into heme-containing proteins, but they have to then take that heme from the intestine, which is the portal for heme entry, and then transport it across the intestine to all the other distal tissues, muscle, hypodermis, neurons, and the progeny, oocytes and sperms. Okay. So here's the first piece of data. If you grow worms under the lowest heme concentration, which is 1.5 micromolar heme, and they take about 9 days to grow a year. Their optimal heme, 20 micromolar, they require about 3 days to grow. Right? So if you grow them under low heme concentration, and you give them a fluorescent heme analog called zinc heme, right? So iron quenches the fluorescence of the porphyrin ring, but you can take the iron and put in other metals, so it still remains as a metallated porphyrin. You can now use it as a fluorescent heme surrogate, and you can look at the heme accumulating within the worm's intestine. And if you grow worms at high heme concentrations, they don't take it up as well, because even though heme is essential, it is cytotoxic. That would mean that their uptake system needs to be regulated. You can quantify this fluorescence intensity here. So here is that heme growth curve that I showed you, different heme concentrations, and here is the fluorescent heme analog. So worms grown under low heme conditions take up heme very effectively. Worms grow at high heme concentrations, switch it off. Tells you it's regulated. So this allowed us to use 3 different approaches, unbiasedly: genetics, 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 right? Forward genetic screens, mutagenizing the entire genome using EMS. Reverse genetics, knocking down all 19,000 genes one at a time, and then doing RNA-seq and transcriptomics, identifying heme-responsive genes. So here's what we have discovered using this is where I use the term, the awesome power of C. elegans genetics, right? The first class of heme transporters, which we named heme-responsive genes, are HRG1s. In C. elegans, you have to follow 3 and now 4-letter codes. So these are heme importers. Since C. elegans doesn't make heme, it has duplicated its heme transporters, HRG1, which localizes to the endolysosomal compartment, 4, 5, and 6. A small peptide that the mother secretes from its intestine, which takes the heme and delivers it to the embryos. An oxidoreductase, which is localized in the hypodermis, and it allows the hypodermis to utilize heme effectively. A heme exporter, which localizes to the basolateral surface, export heme to all the other tissues. In its absence, the mother as well as the embryos are dead. A cell signaling molecule, which we named HRG7. This is secreted under low heme conditions. Internalizes into sensory neurons. The sensory neurons in turn sends a BMP5 homolog called double-1, which turns on the SMAD transcription factors, and it represses this gene. So this is the accelerator, and this is the brake, which allows the entire organism to respond to dietary or environmental heme, because all of it is dependent upon the heme coming from the food through the intestine. Okay. So the ultimate goal for us remains, can we exploit C. elegans to identify analogous pathways in vertebrates, and can we then exploit differences between the human heme acquisition pathways to the other parasites, because as I mentioned before, most parasitic nematodes or helminths do not have the capacity to transport heme. So this I'm going to discuss about one transporter today, which we named HRG1, or now it's called as SLC48A1. So HRG1 was discovered as these proteins and genes that are highly expressed under low heme conditions. They're regulated transcriptionally, post-transcriptionally, as well as translationally. Okay. They are paralogous transporters. C. elegans has 4 of them. These 2 are highly heme responsive, shown here, and 5 of 6 are regulated at a post-transcriptional level. We have, and vertebrates have, only 1 of these transporters. So I'm going to discuss this through many different organisms and what the role of these first class of heme importers look like. So these are 4 transmembrane domain proteins, which is not what I would have predicted. Hemes are approximately 560, 570 Dalton organic cyclic molecules, and I would have thought that these transmembrane domain proteins may have 8 to 12 transmembrane domain proteins. So all I can say at this point is that these are at least a trimer, if not a tetramer. Okay. You can see that this is HRG4. It lacks this green and this blue residues shown in HRG1. And for those people that are interested in membrane trafficking, this green residues are tyrosine-based sorting motifs. The blue residues are dileucine-based sorting motifs. And they're required for retrieval of the protein from the plasma membrane to the endosome, taking endocytic protein to the lysosome. 4 lacks those. The histidine has been replaced by a tyrosine. Histidine prefers binding reduced heme. Tyrosine prefers binding oxidized heme. The human transporter is only 20% identical, but if you look at it, it's still 4 transmembrane domain proteins, but those critical residues that I've shaded in red right here, they're highly conserved in the human proteins. And if you mutate those residues, the transporter is dead. It is unable to transport heme. Okay. So what do these guys look like in vivo? Here is C. elegans HRG1. These are the intestinal polarized cells opposing each other. This is the lumen, dark lumen in between. You can see that HRG1 localizes, and I can say for sure, this is the fluorescent heme analog, and these are lysosomal related organelles in C. elegans. Okay. They completely surround these LROs. And here is HRG1, which lacks those green and blue residues, and they localize exclusively to the apical plasma membrane, shown here, and this dark line in between is the lumen. You can completely change its localization by simply flip-flopping its C-terminus. So its localization is dictated by C-terminus. And here is the humanized worm, where we have deleted all 4 transporters, and we put in the human gene into the worm. The human gene fully rescues the C. elegans quadruple mutant, and more importantly, if you notice, is that the human transporter localizes not only to the apical plasma membrane, but to the endosome. So it's taken over the function of both HRG1 as well as 4. Okay. So the question that we always got whenever we submit a paper to a journal is this: do mammalian cells ever utilize exogenous heme? Because all of the work with your C. elegans stuff only tells you that, yes, C. elegans is a hemoxotroph, and of course it transports heme. Right? So there is absolutely no evidence that heme, as a single molecule, moves directly from cell to cell in mammals, and likely all vertebrates. After heme is made in a mammalian cell, it remains inside the cell until the hemoproteins are catabolized. Right. So I use these kind of reviews as a way to pivot myself and refocus. So the way to address this question was to see, do mammalian cells ever utilize exogenous heme? Now it's been known for at least since the 1970s that patients with porphyrias, these individuals have mutations in the heme biosynthetic pathway. They are not null, otherwise they will not live. They are typically hypomorphic alleles of heme biosynthesis, and these individuals can be treated with heme. So that would mean that cells could probably take up heme if given exogenously. So we decided to address the reviewer's question in a different way. We took 2 peroxidases, horseradish peroxidase, ascorbate peroxidases, but they're both plant proteins, and they bind heme with a relatively decent affinity, Kd of approximately 270 to 350 nanomolar. And then we took those peroxidases and localized it to all intracellular organelles. So here is HRP, localizes to the ER, Golgi, and plasma membrane, and APX, or ascorbate peroxidase, to the cytosol, nucleus, and mitochondria. And we then ask this question, do mammalian cells have the ability to utilize exogenous heme? Right. So now what we can do is take off any contaminating heme, and there is some in the serum in cultured cells. So we can heme deplete them. We switch off the heme biosynthetic pathway using a chemical called succinyl acetone, which blocks the second step of the pathway. And now because it's HRP or APX, you can do in situ, using diaminobenzidine. You can do plate readers, or you can do microscopy, right? So we cannot or barely detect activity in all of these 6 compartments. Switching off the synthesis, you turn on the faucet from the outside. So you bring in the heme from the outside. Anywhere between 1 to 4 micromolar of heme is sufficient to restore heme levels in all of those compartments. So these results say that mammalian cells are quite flexible. They can make heme, but if you switch off heme synthesis, or if they require heme from the outside, they can take it up. Right. So let's go back to this question. Do mammalian cells require a heme transporter? So we made antibodies against the human homolog of C. elegans HRG1, and we found that this protein is specifically and highly enriched in macrophages in the liver, spleen, and bone marrow. And these tissues are called as the reticuloendothelial system. The RES macrophages play a really critical role in iron metabolism. We have approximately 25 trillion red blood cells. Each red blood cell contains about 1 billion heme molecules. And every second, just like this, we are recycling 5 million red blood cells, because the lifespan of a red cell is approximately 120 days in humans, or in mice between 40 to 60 days. So as they undergo senescence, the red cell membrane lipid composition slightly differs. These are recognized by the phagocytic receptors in the reticuloendothelial system macrophages, Kupffer cells, red pulp macrophages. They phagocytose the senescent or dying red blood cells. The phagosome matures into the phagolysosomal compartment. The red cells are degraded and they release the heme. This heme is then imported into the cytosol. And the current dogma is that this heme is then exclusively degraded by heme oxygenases to open up the ring by breaking up one of the methane bridges, releases the ion, releases a mole of carbon monoxide, and makes that linear ring now into biliverdin that gets converted to bilirubin, conjugated, and excreted out. The ion that's released then is exported out by ferroportin, picked up by the transferrin protein in the circulation, taken to the bone marrow, picked up by the transferrin receptor in the bone marrow to make new red blood cells. Or the excess ion gets stored into ferritin. So here is the open question, right? Every second, we degrade 5 quadrillion heme molecules to make 5 quadrillion heme molecules back in the bone marrow for new red cell production. That's the current thinking. And I'll be happy to answer questions for that. So the question we asked was, given the fact that HRG1 is so highly expressed in macrophages, it localizes to the lysosome, and it is a heme transporter. Is it possible that HRG1 is the macrophage heme transporter, which will put it upstream genetically and cell biologically from heme oxygenase ferritin and ferroportin? To do this, we isolated bone marrow macrophages. So that's what this is. And we found out that typically, yes, in bone marrow macrophages, HRG1 localizes to the late endosomal compartment. If you give this macrophages latex beads, HRG1 will go decorate those phagolysosomal compartments. Then it realizes it's been tricked and goes back to the late endosomal compartment. But if we give them opsonized red blood cells, or oxidized red blood cells, those HRG1 will get enriched on the phagosomal compartment, shown here, and they're systematically, continuously being recruited from the late endosome to this gigantic phagolysosome shown here. So is HRG1 upstream from hemoxygenase ferroportin of ferritin? So if you knock down HRG1, we get about 80 to 90% knockdown. And now if you feed them red blood cells by this process called erythrophagocytosis, you can see that during erythrophagocytosis, hemoxygenase, ferroportin, and ferritin goes up. But if you knock down HRG1, all of those downstream markers are reduced. So these results tell you that, yes, cell biologically, HRG1 is upstream from all of these three major heme contributing genes. So we knocked out HRG1 in mice. This was done right here at NHGRI with my friend David Bodine. So our initial hypothesis was simple. HRG1 knockout mouse is going to be embryonic lethal. These mice looked indistinguishable from wild-type mouse when you look at it from the outside, till you open up from the inside. As soon as you dissect the mouse and you open up, all the rib cages are extremely dark. You look at the spleen is dark, the liver is discolored, and this is what a single femur looks like for a knockout. It's really dark, but it's not the bone itself, because if we flush out the marrow, this is a single cell with this dark black pigments within the cell, and here is the marrow cell. So there's something really unusual about these mice. If you look at the three RE tissues, the spleen, liver, and bone marrow, here's wild-type mouse, and here's what the knockout mouse looks like. They have these really dark pigment that appears to have precipitated within these cells, and these cells are macrophages of the reticular endothelial system. So we can take those tissues, grind them up, and precipitate those dark pigment, and the only pigment that was known to occur in living biological systems that we knew of was hemozoin, and hemozoin till then was discovered to be made by Plasmodium parasites. Plasmodium engulfs large amounts of heme within red cells and then sequesters heme as hemozoin crystals into its lysosome called the food vacuole. So we use this hemozoin dissolving buffer to this pellet, which was insoluble. Hemozoin dissolving buffer is simply basic pH with SDS. All right? And voila, you got the pellet is completely dissolved. It gives you the beautiful heme spectra. We can take this pellet and do something called X-ray powder diffraction, and we got back the structure, and the structure is identical to Plasmodium hemozoin. So these results told us that, yes, the mouse appears to be normal because it's taking up all of the heme that's accumulating within these macrophages and polymerizing them into these crystals called hemozoin. This is what a Plasmodium crystal looks like. In scanning EM, here is what the splenic, liver, and bone marrow hemozoin looks like. Even though their surface looks slightly different than Plasmodium hemozoin, they're at the ultrastructural level, they're identical. So here is a wild-type macrophage, bone marrow macrophage, or a splenic macrophage. Here is the beautiful lysosome. This is HRG1, LAMP1 positive compartment. Here is an HRG1 null mouse. These hemozoin have been pseudo-colored from black to red. You can see they're in this gigantic lysosome surrounded by the lysosomal membrane. And these lysosomes are significantly larger in the knockout than wild type. So these macrophages are accumulating heme in the form of hemozoin in this gigantic lysosomal compartment. So how and why do these mice make hemozoin? And what is the basis for them tolerating this high amounts of heme? So to be able to address that, we took a donor mouse, a wild-type mouse, injected them with a hemolytic compound called phenylhydrazine, which causes hemolysis, gave them radiolabeled iron, iron 59. And after 3 days, we can sacrifice this mouse, and majority of this iron radiolabel will get into their red cells. And the hemoglobin now will get labeled by iron 59. So in our hands, about 87% of this iron 59 gets incorporated into hemoglobin. We can take this labeled red cells, opsonize them or damage them, and give them to recipient mice, either wild type and knockout, and then sacrifice the mouse 24, 72, 96 hours later to look at the kinetics of clearance of these red cells. And here's an example, just looking at the spleen. Wild type mice are able to clear most of this radiolabeled red cells. This is all that's remaining after 96 hours. Here's our knockout mice, tons of radiolabeled iron. We can distinguish heme from non-heme iron. Hemes are organic, so they go into organic phase. Iron goes into the aqueous phase. You can see that there is a significant amount of heme stuck in the organic phase in the knockout. But this plus non-heme iron does not equate to that. We are losing somewhere. So if you now look at the pellet, the majority of the iron 59 counts are in the pellet, that hemozoin pellet. You can take that hemozoin pellet and use the hemozoin dissolving buffer, and now this is where the radiolabeled heme is going. And here it is. The majority of the iron 59 is going into hemozoin heme. So why are these mice surviving if they're unable to recycle the heme iron? That's because a normal lion is fed way too much iron in their chow than normal. A typical mouse needs about 20 parts per million of iron. Standard chow has 380 parts per million. So if you now take these wild type and knockout mice and feed them low iron diet, which is about 2 parts per million, both the wild type and knockout mouse, the hematocrit drops from about 45 down all the way to 20 for the first 5 weeks. Here is your control. They remain not significantly different till after 5 weeks. After 5 weeks, they reach this new steady state level, okay, where they're solely relying upon recycled iron, whereas the knockout, because it's unable to recycle iron, continuously accumulates hemozoin heme, and eventually they succumb because of severe anemia. So there is massive changes in gene expression between the wild type and knockout mouse if you look at the gene expression profile at 5 weeks. Okay. So the model for how does HRG1 knockout mouse survives and is able to tolerate this high amounts of heme, as I said before, body iron is a composite of the amount of iron coming from food versus the amount of iron that's being recycled by the 3 major organs, spleen, liver, and bone marrow. The main bone marrow produces new red cells. In the absence of HRG1, there's no recycling going on from the spleen, liver, or marrow. Spleen starts accumulating hemozoin, liver starts making hemozoin, and the bone marrow starts making hemozoin. Eventually, you'll end up in something called ineffective erythropoiesis. So the bone marrow says, hey, spleen, could you give me a hand? The spleen tries to expand, but it's unable to undergo stress erythropoiesis to help maintain normal hematocrit. And eventually, these mice start developing large amounts of hemozoin in their spleen, liver, and marrow, and their organs really are messed up. In the case of liver, there are large lipid droplets within parenchymal tissues. These mice continue to survive even though they're unable to recycle because they're still getting a lot of their iron to be able to make new red blood cells. Okay. However, if you remove the iron source, as we did under low iron condition, these mice die. Okay. So I'm going to switch gear in the next 5 minutes to discuss this angle about how do parasites acquire heme. So as I mentioned before, heme biosynthetic pathway is more or less highly conserved on this planet. We have shown that parasitic helminths, including, of course, C. elegans, which is not a parasite, lacks the ability to synthesize heme, but hijacks heme from the outside. So what does that mean in terms of an evolutionary perspective? Would that mean that the ancestral worm or the ancestral organism was a parasite that was living in a heme-rich environment such that it said, why do I need to make it when I can eat it? Or is it possible that it was living in an endosymbiotic relationship such that the endosymbiont was producing heme in exchange for for nutrients from the host. While both of these scenarios could be true, hookworms live in the intestine and gorges on huge amounts of blood. And Wolbachia, which is an endosymbiotic bacteria that lives within Brugia malayi, and if we knock out the heme synthesis pathway or block the heme synthesis pathway in Brugia malayi, in Wolbachia, the Brugia malayi dies. None of these organisms are genetically tractable. To be able to get to a genetically tractable organism, we started working with a parasite called Leishmania. Leishmania is a trypanosomatid. It lacks the apparatus for heme synthesis. And the most interesting thing for us for Leishmania is that it can go through various different differentiation stages. It is transmitted by flies, so it has a procyclic and metacyclic promastigote stage. Once it's injected into the host, these metacyclic promastigotes invade the macrophages of the reticular endothelial system in the case of Leishmania donovani. They live inside the phagolysosome of the same macrophages that are recycling all of the heme and the iron. And these amastigotes continue to replicate inside the macrophages until they cause demise of the host red cells. New amastigotes are released, and they infect neighboring cells. Okay. The beauty of Leishmania is that they are genetically tractable. You can knock out genes. You can knock in genes. You can resolve them by growing them azenically in liquid culture. You can infect macrophages in cell culture to look at host-parasite interactions, and you can do in vivo experiments within genetically altered mice or hamsters. So by using some interesting bioinformatics, we were able to identify the first parasite heme transporter. So we just named them Leishmania HRG1 or LHR1. Leishmania is a diploid. So if you make a single knockout, also called as heterozygous in mammalians, the amount of heme drops by half. You cannot make a double knockout, which will be homozygous, because it is lethal. You can put in a transgene and it can restore heme levels almost as good as wild type. Here is a single knockout or heterozygous Leishmania for LHR1. It is unable to form lesions in mice compared to wild type mice, and here's the genetically rescued. Okay. So heme regulates LHR1 and HRG1 completely differently. In the absence of heme, LHR1 is upregulated. This parasite cannot make heme. It absolutely requires heme to survive, so it has to hijack heme. So the protein is turned on in the absence of heme. As heme concentration goes up, it dials it down. Unlike that, human HRG1 is turned on when there is heme, as you would expect when it's required to undergo erythrophagocytosis. So the dynamics and regulation of macrophage HRG1 and host LHR1s are quite different. So here it is. This is human HRG1 or host HRG1 on the phagolysosome of a macrophage. These same macrophages, or reticular endothelial macrophages, are infected by Leishmania donovani. It's a heme auxotrope. It requires heme for its own survival. It has a different variation of the same 4 transmembrane domain protein. It's trying to hijack the same substrate or nutrient to be able to replicate and divide. The interesting thing is that LHR1 and HRG1 are only 13% identical, which has implications for the next slide. So the question we are asking is that does this competition or heme tug of war occurs in the same intracellular compartment? So this is the last 2 slides. So we have done a high throughput drug screen to be able to identify compounds that specifically block the Leishmania heme transporter without touching the human transporter. And we have identified really interesting scaffolds, chemical scaffolds, that are able to antagonize LHR1 without touching the HRG1. Okay. And here is an example of this. So these are 2 compounds They're working in submicromolar concentrations to target LHR1 within the host macrophage without killing the host cell. So ultimately, there are some really outstanding questions that we want to answer. These are not outstanding as in amazing questions. These are outstanding because they still need to be answered. How is heme homeostasis regulated at the organismal level? We have been able to get some information at the cellular level and some information at how multiple cells communicate. But how does the entire organism deal with this? Is there a heme sensing mechanism? What is the pathophysiological role of HRG1 in humans? We are still looking for human mutations that are homozygous for HRG1 if they are there. How is heme acquired at the host-parasite interface? And how does heme traffic in living cells and early embryos, which we really don't understand? Okay. And we are addressing it by using genetic screens in C. elegans to be able to look at how organs and tissues communicate. What is the pathophysiological role? Are there human variants which are genetic modifiers of hemolytic diseases, such as sickle cell, thalassemia, plasmodium infections? There was a disease that was described over 100 years ago called African iron overload, also known many years ago as Bantu cirrhosis. Is this the genetic etiology of Bantu cirrhosis? How is heme acquired at the host-parasite interface? And can we use small molecule inhibitors of HRG1 and LHR1? And finally, can we devise heme sensors and non-invasive label-free imaging to be able to look at heme as it's moving on between cells and within cells? With that, these are the amazing group of people that have been doing all the work. And one thing you'll notice from phenomenal set of collaborators, the number of people that have collaborated at the NIH. Right? They have been very generous with their time, with their resources, and I've deeply appreciated their help over the last 20 years. And with that, I'll be very happy to answer questions. Thank you for your time and attention.
So, Iqbal, that's great as usual. Always learn a lot. So, if I remember that slide with the Leishmania, if you put that Leishmania into your HRG1 minus mice, where the heme is all polymerized in hemozoin, does that make them resistant to Leishmania?
Okay, so Dave Bodine asked me this question that if you infect HRG1 knockout mice, which is accumulating a ton of heme as hemozoin, with Leishmania, what would happen? So my graduate student Melissa Perry has been doing these experiments, and the answer is that they have very low parasite load. Now, if you take the single knockout parasite, which is quite crippled, and you infect them, they do even worse than wild-type parasite. But if you look at their organs that are infected, they're much larger than wild-type. Typically, when you have splenomegaly or hepatomegaly, it typically is a sign that they have more parasites. But here, it's the opposite. You do see hepatosplenomegaly in the knockout when you infect them with Leishmania, but they have exactly opposite outcome. And we are trying to figure out why that is the case.
Yeah, very interesting talk, Iqbal. Thank you. I was curious in thinking about mammalian cells. Do you think that they are making a choice about whether to make it or to import it for their use, and if some cells have a preference? Or do you think it's just evolved as a protective mechanism against excess heme that they can do it?
So Ashley Frakes, I'll eventually come to a point where I may not know the investigator. Ashley Frakes asked me this question, that do mammalian cells make a choice that they can make their own heme when they want to, and where they can take up the heme from the outside when they have to. So I'm going to answer this question in collaboration with my colleague Makiko at Mount Sinai. We knocked out the heme synthesis pathway in hepatocytes. This is unpublished, right? The initial thinking was heme synthesis is so crucial that if you knock out heme synthesis, the cells should not be able to go through one round of replication. But when you do it in vivo using LOX-P and Cre excision, and we knock out heme synthesis, they have completely normal heme in hepatocytes. Okay? And I'm going to make a long story short of doing a series of experiments over the last 2.5 years. The short answer is that they get a significant portion of their heme from Kupffer cells. Okay? So I have evolved with this thinking now in the last 6 months that mammalian cells are quite plastic in their heme quota. If they need to make heme, they will make it. And if they need beyond a specific threshold, they'll get it from their surrounding cell. And what better cell to get it from than a professional heme recycling cell, such as a macrophage. So in any sense, I think that this is evolving into something which C. elegans has decided a long time ago. Why make it when you can eat it?
Yeah, thank you.
Well, if there's no more questions, let me repeat the CME code for the folks playing at home. Just a minute. You're going to have a question? Okay. The CME code is 44431. Okay.
So Iqbal, I had a question about sort of in general, so either upstream or downstream of transport, heme looks like a molecule that would need to be chaperoned. I'm just curious, do you think that we have a sense of the gamut of chaperones that cellular systems utilize, or do you think there are going to be surprises there? I'm just curious to know your sort of general thoughts about this.
Do I have a sense of the gamut? I don't have a sense, partly because I wouldn't have the sense 15 years ago to predict HRG1 as a heme transporter, right? It came out from our genetic screens. But does it make biochemical sense that they should have one? Absolutely. And I give this example a lot, actually, that when cholesterol synthesis pathway was systematically dissected, people said there is no reason for cells to transport cholesterol. Since then, investigators have identified quite an intricate network of cholesterol transport pathways. As I would consider cholesterol to be quite innocuous compared to heme for its cytotoxicity. And cholesterol, if you knock out the protein, which will be almost equal to HRG1, called NPC1, accumulates cholesterol within the lysosome of macrophages. Within the lysosomal lumen contains NPC2, which is cholesterol chaperone, which takes up cholesterol and delivers it to NPC1. So if a molecule like cholesterol can have a chaperone and a transporter, you would bet that there would be a heme chaperone within the lumen of the lysosome to be able to pick up the heme, deliver it to HRG1, and that heme then should be relayed from HRG1 to any of its downstream effectors. So we are doing that in vivo using this HRG1 GFP mice that we developed in the last 2 years.
The question is about the experiment you talk about where you knocked out the heme biosynthesis pathway in the hepatocytes. So is HRG1 upregulated there, and is HRG1 going to the plasma membrane, or there is another transporter that brings in the heme into the hepatocyte?
Into hepatocyte. So Wing asked me this question, that if you look at the hepatocytes that has now that cannot make heme, is HRG1 upregulated? I don't know the answer to that because we just got the single-cell RNA-seq results from those. But all I can say is that HRG1 from macrophages is important for hepatocytes to get its heme. I do not know whether HRG1 from hepatocytes is important or not. And the only way to do that experiment now is to knock out HRG1 in hepatocytes and then do the bone marrow transplantation experiments that we did in this hepatocyte-specific knockout mouse.
Yeah, nice talk. Can you comment on the mechanism of heme transporting?
By HRG1,
Yeah.
I can hand wave, and I can say this, and that there is a histidine in the extracellular loop, which is critical for transport. So the heme goes from extracellular lumen into the cytoplasm. There is a histidine in the transmembrane domain. If you knock it out, the transport doesn't happen. And there are 3 residues in the C-terminus tail, which is essential for heme to get out. Let's call it a channel for the sake of anything else, to get out of the channel into the cytosol. It is coupled to a V-type ATPase. So if you knock out the V-type ATPase, then HRG1 can no longer transport heme. So it's a proton-coupled secondary transporter. I do not know anything about what its structure is. On a blue native gel, it runs minimally as a trimer, but it could be a tetramer.
So you are targeting that histidine domain
I,
with your inhibitors. Can you also comment on your how does your inhibitors look like?
God, chemically, I can't even begin to tell. There are 3 or 4 ring structures. They have not been used in any previous drug discovery. They are quite novel. I've been working with a medicinal chemist to be able. All I can tell you is this for a fact, that they antagonize HRG1 and they block HRG1 or they block LHR1. Okay. Our initial goal is, I'm going to finish this by saying that if organisms can really tolerate 10, 20-fold high heme by simply sequestering them into hemozoin, it is possible to use antagonism of HRG1 as a way to overcome hemolytic crisis.
Great, thank you.
Okay. Yes, we do have one from our colleague, Alan Scher, a self-described basic question. What is the basis of heme toxicity, and why has the body chosen to transport iron in a toxic molecule?
Why has the body chosen to transport iron
In a toxic molecule.
Oh, I see. Iron within heme as a toxic molecule. Okay. So iron sulfur clusters, copper, and heme. So if you were to look from an evolutionary perspective, hemes have evolved much later in life through evolution than iron has, iron-containing proteins, or copper-containing proteins that are involved in oxygen transport. Crabs and lobsters don't have hemoglobin-based oxygen transport system. I'm looking at you as if you're Alan Scher.
Yeah, no, I'm trying to play the part as well.
So I think that the reactivity, as the oxygen concentration increased in the atmosphere, hemes were shown to be far more catalytically superior than other forms of metallo cofactors.
Got it.
So why has the cell chosen to transport heme instead of non-heme-based cofactors? I can only hand wave.
Okay.
I don't have a full answer for that, Alan.
Well, let me conclude by saying thank you that you've inspired at least a dozen people to join the WALS listserv in the past hour, which is excellent. And Dr. Banerjee, do you have any closing thoughts?
So with that, let's thank Iqbal for a fascinating